It is time to get this party started again.
Hi, Ross.
Hello, how are you?
Doing well. How are you doing?
I'm good. Are you ready?
Yes, I'm ready. Um,
Excellent. So I just want to say a couple words to introduce Laura Towart. Um, I'm really excited to have you come and speak to this group. I hope you've gotten a sense so far. It's a pretty active group. You can
yes.
expect some pretty heavy questions, but I suspect you can handle it. So Laura has been a true pioneer. I think I can say that pretty clearly as a fly entrepreneur among entrepreneurs in non-fly stuff. You started your second company, is that correct?
Yes.
Yeah, so very impressive. I'm not going to gush on for 10 minutes about you. I'm going to give you some space to give a talk. So I hope everybody enjoys this. I think you really will. So Laura, please take over the screen.
Okay, thank you. Okay, well, hi everyone. Thanks for this opportunity to share some more about myself, my venture, my personal therapeutics, and our journey using Drosophila as avatars for personalized medicine for cancer therapeutics. So a bit of background on the technology and the process of commercialization. I was introduced to Ross about a decade ago and was immediately interested in the potential for the personal discovery process technology and its potential for personalized medicine, originally for colorectal cancer. And so first, the technology was developed by Ross and validated at Mount Sinai for use in patients, supported by NIH grant funding, and then ultimately licensed to My Personal Therapeutics for global commercialization. This happened mainly around 2019. So to understand why the technology is so important, I'll just share a bit of background about cancer treatment, as many of you may know. So up to 75% of first-line cancer treatments fail, and oncologists don't really have any opportunity, but don't have any other alternative but to treat by trial and error, trying one targeted therapy and then seeing if it works, and if it doesn't, trying to identify second line or third line. So whereas advanced flight simulators allow critical decision-making to be learned safely, our cancer simulator using flies for oncologists can eliminate dangerous trial and error therapies on patients. And we're also using it to power biopharma. So prospective drugs work successfully in preclinical settings, but we know that over 90% of them fail in clinical trials. And so as Ross has stated, these drugs seem to work beautifully on mice in the lab, but they don't work on patients. And the question is, why not? Each cancer patient has over 100 mutations, but we can't represent multiple mutations in mice or many mutations in mice. And so the fly is realistically the only animal that can simulate the full complexity of cancer. I know I'm probably preaching to the choir here. So at My Personal Therapeutics, we harness the full complexity of biology to give oncologists and pharma the most powerful therapeutics decision tool. So the personal discovery process technology replicates a patient's genetic profile and enables us to screen thousands of drug cocktails. So step one, a patient would have their tumor biopsied at their point of care and then sent for whole exome sequencing. And that's really where our process begins. We do a deep interrogation of the tumor exome, and we identify the mutations that are seemingly driving tumorigenesis. And we can select up to 20 mutations, and we engineer them into the Drosophila, creating avatars. Then we grow up about 500,000 avatars or flies, and then that enables us to do high throughput drug screening using all FDA or in Europe, EMA approved drugs. And then we screen them in combination. So tens of thousands of drug combinations. And then we see which drug cocktails rescue the flies. And then we make a human treatment recommendation. So, well, Mount Sinai has had success in using the technology to treat the very hardest to treat patients, even after multiple other treatments fail. So one patient, this is not his photos to representation. He's a 53-year-old man, had KRAS positive colorectal cancer, and he had failed every other treatment and had no other treatment options available to him. So his tumor whole exome sequencing and analysis identified recurrent KRAS, APC, 253, on a number of other mutations. And so a personalized avatar was created for him, including 9 of these. So after drug screening, a novel combination was identified and the patient was treated. You can see here his CT scan. He had in red is the target lesion. And you can see that after 27 weeks, this target lesion was reduced by 45%. And this study was done with Ross and Dr. Banji and was published in Science Advances in 2019. So at the time of this publication is really when the company's commercialization began. This paper enabled us to initiate fundraising and really be able to demonstrate that to investors and to really the world that the technology was powerful, even if it was only in 1 patient so far. And we raised funds in order to really expand operations and be able to offer this to many more patients. So we call our scalable in vivo model, it enables us to do what really no other model can do, creates a virtual biological twin of the patient, which can replicate the full tumor complexity in a living biological system instead of petri dish or even an organoid, and rapidly test thousands of drugs and combinations in many, many avatars, enabling great statistics. And then having treatment recommendations be combinations that incorporate non-cancer drugs, making treatment potentially less toxic and also more affordable overall. So what we've done is we've started to take a lot of the data generated by Ross and Mount Sinai on patient whole exome with the avatar created, and then with the corresponding drug treatment recommendations, and really start to compile all of that data. So that's what we call our proprietary data set, combine it with some other omics data sets, and then use that to make a biological twin simulation engine, which is kind of our fancy word for using deep learning to power a virtual lab space and an experimental lab space for pharma, and also to power decision-making for oncologists. And we can do this because we know that patients with similar tumor genomic profiles will respond to the same drug treatments. And so we're in the process of building this now, and it's really important because we want to be able to decrease the time that it takes to give patients treatment recommendations. So we face many, many challenges along the way, and we still face challenges daily as a venture. Funding is always a big challenge, and especially when you're, the flies aren't as fancy as organoids or some other new technologies that are out there. So we face, a hurdle by bringing something very old school to the investment community and saying that this is actually really exciting and actually very high tech. It seems very low tech when you're talking about fruit flies. So funding is difficult, especially in the face of COVID. Building the right team and kind of the human component is also difficult. Trying to find the right specialists and bring everyone together to work on a common mission. One problem we face today is that there's not a lot of clinical data. And so as a company, we do need to have oncologists recommend this to patients. And there's the problem that there's just not a large amount of data. And it does take time to also generate data, as it takes time to build each fly model and do drug screening. And so COVID, again, has been a really big issue, not only just for funding, but because as we started to generate traction, a lot of patients, unfortunately, their treatments were halted. This happened, I think, many places around the world where patients couldn't get tumor biopsies. Anything that was kind of non-critical wasn't permitted. And unfortunately, that extended to cancer patients in treatment. And so also one of the other issues is that treating patients is time dependent. A lot of patients that come to us are patients that have run out of options. They don't really have the time to wait 5 or 6 months to get treatment recommendations. So we're working on fixing that problem by introducing, AI or deep learning, and then also trying to improve the drug screen strategy. So this is kind of illustrating, This is illustrating, the stages of life cycle of the Drosophila, and this is really where we're doing the screen. So the patient avatars without the correct drug, will die sometime during these larval stages. And so right now, scientists actually have to count the number of surviving flies and distinguish between the experimental versus the control larvae. This does take a long time. So we're in the process of working with a company called Labman to build an image capture system that could help us speed up the time. So Labman, the system includes a camera mounted into a gantry arm, which would pick up each screening vial, place it on a rotary stage, and then have a 360-degree camera that would take images and then stitch all of the images together. So then we engaged another company called V7 to assist with image analysis, annotation, and also model building. So the model is really a neural network that would automate the process of identifying the pupa. And then the next phase would be deployment, where we can actually utilize the system to decrease the time it takes to gather the screening results and ultimately deliver treatment recommendations to patients. So an effort such as this wouldn't be possible without the determination and dedication of our team. Our scientific team is led by Dr. Nob Hofelegas, who has years of experience as a Drosophila biologist. He's also engaged in drug screening. John Waterman-Smith, who's our VP of Business Development. We have a dedicated CTO, Paul Cook, and then we recently hired Dr. Eduardo Abini, our bioinformatician, and we have also now a team of 11 full-time employees, many of whom are scientists. So we are also supported by a world-renowned advisory board, including Ross Kagan, Dr. Elizabeth Smith, Boaz Benali, Michael Elishoff, Michael Castro, and Meher Gulshan. So all leaders in oncology, pharmaceuticals, and also machine learning. And we're partnered with oncologists at over 15 medical centers around the world, each treating hundreds of patients annually. And we're always looking for additional medical centers to work with and collaborate with, and explore even new avenues other than, we right now we're really focused on GI cancer and lung cancer, also rare genetic cancers, but we're looking also to expand into other areas like glioblastoma, also looking at diabetes and cancer. So we're lucky enough to have received funding and support from the investment community and also Innovate UK, Eureka, and the Wellcome Trust. We've gotten a lot of interesting awards and recognition over the past 2 years, and we really find that people are very interested and excited about the technology and really its promise for cancer and other genetic diseases. As our approach is very unique, we've also generated a lot of interest from the media and other publications, both scientific and otherwise. And we really are very fortunate to have the support of such an active fly community. And we hope that together we can make a real impact and continue to use the flies to simulate genetic disease and stop experimenting on patients. Thank you. So I'm happy to answer any questions.
Excellent. Thanks, Laura.
Thanks.
And thank you for featuring some of the work we've done in the lab. But I do want to point out to everybody that Laura and her group are really taking this technology much further forward. The AI and so on is really brilliant. I love it.
Thank you.
I think it's clearly the future to drive this and make it more useful. So we have questions. From Agojo Oilbo, remember, got to give me a break on my pronunciations. It's really great to know we can use flies in personalized medicine. And I'm going to take that and push that now towards a question, which is that, what other, you touched on this a little bit, what other options do you have? Other, animal systems, why not use mice for it? Why not use yeast for it? Yeah.
Sure. And so, mice are very useful in studying cancer and other genetic diseases. One of the issues is that you can't introduce many mutations into mice quickly, and really you can't introduce many more than 2 or 3 at all. So it's just very difficult. So we're using kind of the power of the flies and the tools that have been developed for the past decades to be able to introduce many mutations to really be able to mimic the real true complexity of a human tumor.
Yes. Well, I would agree with that.
So
So how many, so what question, of course, that's going to come up a lot is how do you know what the drug dosing is, how and its formulation, and how do you deliver the drugs to the flies to do the screening?
Sure. Okay. Well, the dosing is a bit more difficult. So we're using, well, we're relying on the oncologist to really figure out what's the appropriate dosing. We're just recommending the names of the drugs and the combinations that we identify, and leaving the actual dosing to the doctors. But I'm sorry, what was the second part?
And how do you deliver it to the flies?
Oh, and so the flies are actually fed the drugs through their food. So they're kind of eating the drugs and the food, and it's there's quite a precise recipe for the food that's very important. And so luckily, one of Ross's students was in the UK before and had solved the problem of the recipe for the food.
Ismail Adinaya wants to know, oh no, I'm sorry. I'm getting myself confused. Agojo Olbo wants to know, how long does it take to screen the drugs with the flies? Because most cancer patients, especially in Africa, only get to know they have cancer at later stages.
So it is taking us some time. We actually we expected it to take maybe only 4 to 5 months, but it's actually taking us a little bit longer. And I think it really depends on the complexity of the mutations that the patient has. So our first patient that we had at My Personal Therapeutics, patient from Japan, had a very complicated tumor, and we send out the mutations, and basically we designed the construct, and it's built at another company. And there was some delays with this. We're not sure if it was because it was very complex in itself, or if it was also due to COVID. So we're trying we'll be able to see over the next couple of months with the future patients how long it will take, but it still will take some time. It could take, at the worst, up to 7 months. But we're really working hard to try to reduce that time.
Well, and the more patients you treat, the shorter that will get, yeah.
Yes, yeah, and that's where the deep learning or kind of AI comes in.
Yeah, which I think is brilliant.
Thanks.
So how do you, so Ramado Bello wants to know, how exactly do you monitor cancer drug efficacy in Drosophila?
So we don't really monitor the efficacy. It's really kind of a fast screen. So the flies are fed the drugs, and then quite soon afterwards, where the flies are counted to see the reaction. So the flies that aren't receiving the right combination won't survive. Only the ones that do will survive. So we, through this counting, we develop the ratio of kind of alive versus dead, or survive versus non-survive, and then come up with a treatment recommendation based on that. And we share all of that information with the oncologist on, let's say, the top 3 combinations, and we give that ratio of survival versus non-survivors.
Which is a good quantitative one. Let's see, Mariam Denike Saladin, can you shed more light on how you guys went about discovering MEK1 and bisphosphonate as the drug of choice for the KRAS cancer man?
That's probably a question for you. So that was your patient.
It probably is. I don't want to take over. Yeah. So let me just say that it's pretty much exactly what Laura said, which is using robotics-based drug screening. So you can grow flies in little tubes or plates, and you can use robots. And when I say you, I mean, that includes me, but also Laura. You can use robots to do high throughput screening. And the key answer here, which also is why an answer investors don't love, which is we don't pick out the hits because we think they're interesting. We just go with whatever the fly tells us works. So a MEK inhibitor and a bisphosphonate worked in that patient's fly avatar, and that's why we used it. I wouldn't have predicted that beforehand, but there it is. Simon Amali says, how do you establish cancer from a patient in the fly for personalized treatment?
Sure. So we really have been focusing on GI cancers and lung cancers. And so, for example, for GI cancer, we select the mutations that we'd like to model. We design a construct, basically, it has a kind of bacterial backbone, and we have it synthesized and into a plasmid, and then it's micro-injected into the fly embryo. And it's cool because we can direct the tumor to grow in the area of the fly that corresponds to the human. So for example, for GI cancer, it's the hindgut. And so then when the fly emerges, it will develop a tumor in that area. And so that's how we call it an avatar.
Well, and then following up on that, of course, there are multiple questions about other tumor types. So Abu Bakar says, excellent presentation. How do you evaluate liver cancer using the flies? And Amina Busola Olarukuba says, this is truly amazing. Can this model be used for breast cancer?
Sure. So we haven't tried liver cancer yet. I'm not sure if it's possible or if it's a suitable model. That's something that we'd have to explore. Breast cancer is a little bit more difficult because if it's a hormonally driven tumor, the fly might not be the best model to mimic it. But I know that Ross had a student that was developing a model for triple negative breast cancer, which I think it is a suitable model for.
We submitted that paper yesterday.
Okay, that's great. Congratulations.
Really cutting.
Yes, breaking news. The flies can be used for breast cancer.
Yeah. So there are, oh my God, there's so many questions. Let's see. Well, one question is, can you share the food recipe on Slack if there's no patent issues? In general, you want to talk a little bit about what one has to think about in terms of patent issues and things like that. This is something that we as basic scientists don't often think about.
Sure, and that's something that there is a lot of question about in terms of how patentable the technology is, and so Mount Sinai and Ross did apply for a patent. The patent hasn't been granted yet, but there seems to be I guess an open patent territory for it. But it's something that really needs to be considered if you have a technology. If you identify something that you think could be useful for the world, it is good to patent it and protect it if you want to develop it, because it's very difficult to develop any technology without a patent, because really investors aren't going to support it. So I think even if technology is patented, it doesn't necessarily mean that it prevents others from using it academically or for research, but it's to protect the commercial interests, which are important, because nothing that isn't commercially viable, it won't get out to the public.
Yeah, you might want to elaborate just a moment on that. So why is it so important from a practical standpoint to have a patent on things?
It's important to protect the space for yourself, and also in order to get funding. So if you want to take something forward, if it's not patented, you won't be able to get investment support for it, because investors aren't in the kind of the benevolent world. They want to invest to make money, and so if there's no potential space that they can carve out for themselves, then there won't be any, you won't be able to bring it forward. So it's really, it seems very, very corporate, but it's that way for a reason, that you need to separate and distinguish what each corporation has as their own intellectual property in order to get funding and move forward.
Nareluda Matlouthi asks, can you, so now I think the questions are beginning to broaden a little bit into, of course, more of the basic science. How can you use these complex avatars? For example, they're asking about looking at the relationship between microbiota and cancer for Drosophila, looking at signaling pathways. I've got numerous questions here, sort of bringing these avatars back into the lab and exploring. What is your thoughts about the advantages and maybe disadvantages of using not a standard 1 or 2 hit model, but really complex models and exploring basic biology of cancer?
Sure. Well, building single mutation avatars versus more complex avatars enables us to really study, downstream effects and really distinguish pathway effects. In terms of the microbiome, I think it's something that we've been very interested in, is how can we make avatars even more personalized? And I think especially when we're talking about, GI cancers, the microbiome surely is going to play a role. That's something that, from an R&D perspective, it's very interesting. I myself am not a Drosophila biologist, but that's something that, when we think about, expanding to the next, personalized frontier, that's definitely where the future is.
So one last question, and then I'm going to post the rest on Slack from Adavin Bismarck Dakura. Because I think it's a really interesting one, and I know it's something that you've had to apply a lot of brain power to. Do you select the mutations for modeling, or do you model all the mutations in the tumor? And if you select the mutations, what are the criteria for selection, and how do you handle issues of bias if you select the mutations?
That's an excellent question, and that's really the million-dollar question, because if you don't select the right mutations, you're not going to have a truly representative tumor. And so we have a ranking system, and we're still in the process of, of kind of optimizing it. And I think with each patient, and we're still in somewhat of an there's always a continuous R&D process and as the world literature progresses, we're able to take information from others about which mutations are actionable or which mutations are involved in different processes. But we do look at, obviously, the known oncogenes, and if it's a KRAS is there, we'll put it in. But if when we start to look at other maybe passenger mutations or other mutations that aren't necessarily they wouldn't appear on a Foundation 1 panel, but we know, or we have some information that suggests that they might be involved in tumorigenesis, we'll add them in. But the selection is very important, and it's something that's still evolving. But there is a basic framework on kind of ranking mutations and their importance towards contributing to tumorigenesis.
Sure. Listen, Laura, many thanks. That was
Thank you so much.
really interesting. Of course, I like the science, but I think it's a beautiful job of some of the things that you have to think about when you try to bring Drosophila into biotech.
Yes, and I hope other people, can continue to, commercialize Drosophila and think about, how we can advance, the future for personalized medicine together.
Definitely. It's probably the place of arguably of largest growth for Drosophila.